It's great to see these depth of questions. I think hopefully we'll have lots of stimulation of questions after the talk. So you can see 20 minutes goes by in a flash. So without further ado, I'll introduce the next speaker in this session, Matthijs Verhoog from University of Cape Town. And the title of the talk today will be Human Neurons Undergo Protracted Functional Maturation into Adulthood. Looks like one of a series of 2 talks on sort of developmental trajectories of human neuron intrinsic properties. So please welcome our next speaker.
Thanks for the introduction, and good morning, everyone. It's great to be here and see many faces again. Can't believe it's been 2 years since the last meeting. The talk I'm going to give, indeed, is going to look into the work we've been doing on the maturation of human pyramidal cells. I actually showed you part of this data at the meeting 2 years ago, but since then, there's been a lot of analysis, and I think the story has matured a bit more. Okay. So, as you know, the human brain has this extraordinarily extended period of brain development, and humans seem to be sort of an extreme example of the general rule that the larger the brain and the higher the cognitive capacity that you aspire to, the longer the developmental period required. So there must be some very important and interesting things happening during this extended period, processes that take time for you to develop these cognitive capacities. And actually most of these processes will be happening after birth. So when humans are born, they have about a quarter of the final brain size, and in the years that follow, the brain will mature. You've got outgrowth of the cells grow, outgrowth of axonal connections, spines are formed and eliminated. And interestingly, all of this on a very slow and protracted timescale. So what was recently shown was that this actually may be intentional, that the pace of maturation seems to be governed from the bottom up. So it's within the cell, the genes that govern like the maturation of neurons, like the outgrowth of dendrites, the growth of axons, seem to be packed away behind epigenetic factors that act as a barrier to actively slow down the maturation of neurons. Now, interestingly, this also holds for the genes that are involved in physiological maturation. And indeed, if you take away these barriers, they showed this in these cultured neurons, then the physiological maturation of the cultured neurons actually sped up. So there seems to be this intentional slowing down of the whole process. And particularly on the physiology side, that gives rise to the question of what things may be happening sort of concurrently on the electrical side of things, like how when basically the functional maturation of neurons and circuits. Now, since my last talk, we've seen a nice publication from the Tamas lab documenting these changes in physiological properties that occur over the lifespan, and also between like big changes in different stages of childhood. So we wanted to know, like, what are the real trajectories of physiological maturation? Like, when is a human neuron mature, for instance? And how do these relate to the ongoing brain maturation? And ultimately, then, how do these compare to mouse, which is a species where most of our developmental work is currently taking place, but which has a completely different rate of brain development. It's really geared towards fast maturation. So to investigate this, we did the usual human brain slice electrophysiology work. We collaborate with these 3 hospitals that gave us access to tissue across the lifespan. In our case, in this data set, that's from 10 months to 55 years. It's exclusively temporal cortex, to sort of really focused on one brain area. And then we recorded from these supragranular pyramidal neurons, subjected them to like a range of stimuli, And importantly, after recording, it was important to always retrieve the cell and measure the depth, because there's been a lot of examples showing that the physiological properties are very closely related to the depth in the supragranular layers. So this is the whole data set here. Each dot is a single neuron. You can see this as a bit of a gyrus here. And we actively try to sample across the layers in each case. So what we found was that many of the physiological properties, the really basic physiological properties, took a very long time to mature. So this is for resistance. Each dot there is a single recording, and this is the GLM fit to that data. And you can see that we start at the resistance of around 100 megaohms, and that sort of decays down to half that value over the next 2-3 decades. So this also held for other physiological properties like membrane tau. Brian just introduced SAG nicely. So interesting here is that SAG ratio seems to be quite little, so the expression of HGN channels seems to be quite limited in the pediatric cases. Seems to pick up only later in life. But also like Rio-based, the amount of current you need to fire an action potential, also this slow development. And finally, in the realm of the action potentials, we didn't really see much changes in the waveform of individual action potentials, but if you fire them in a train, that's when a big difference became apparent. So in adults, if you fire a train of action potentials, that waveform is very stable. But in kids, you can see that the action potential waveform really broadens out, so it's like it's not able to maintain that shape very well. So that adult ability of having a very stable waveform is also something that's late to develop. Now, protracted physiological development. Now you also notice that there's quite a lot of spread in the data. Now this is partly, of course, because of our sampling regime, like going across the layers, so we're introducing some variation there. So like in these models, we always take somatic depth into account as a covariate. But even accounting for that, there still seems to be big changes in variance over with age. So for instance, and this seemed to be variance within subjects, so not between subjects. So what I mean is that, for instance, for an infant of 1 year old, the neuronal population will have a higher resistance, but also a much wider spread in resistances, which gives the interesting situation where you can have like very immature cells in terms of resistance, sort of intermixed with ones that look way more adult. So you can see like, for instance, you've got some very young cells already having like a 25 megaohm input resistance. So then, compared to mouse, we have a similar data set, and we see very much the same thing. I mean, the scale of change is very, very different, but the nature of the change, I would say, is very similar. It's just that the time scale is vastly different. Now, in principle, this idea that the time scale is different is not very surprising. I mean, we already know that the human brain is much slower to develop. So the question is really, is this proportionately slower or not? So to investigate that, we looked at we basically normalized all those developmental curves that we fit and looked at them in the context of brain volume over the same period. So you see these are all the different fits, and on top of that you see the change in brain volume over that same period. And you can see that in mice this seems to be very closely linked. However, in humans, that seemed not to be the case. So in humans, they seem to follow a much more protracted timeline. So next, we wanted to sort of put a number to this. I mean, apparently it's not uniformly scaled up. So how to sort of quantify this better? And for this, we turned to some dynamic time warp analysis. This is a technique where you can basically take two time signals and see how much you should warp one to become the other, which basically gives you the ability to match the time points. So, for instance, these two brain volume curves, we now can link every mouse day to a human year in this case. And the result is this warping path, that's the gray line going up there. And you can see that this is pretty linear. which means that we can basically scale one brain development curve to become the other with a single scalar, which is 162 days in this case. In one day, a mouse achieves the same proportional brain growth as a human does in 162 days. So if you take that same approach, but now on those trajectories that we fit, that also gives you these linear warping paths. So the shape of the development seems to be very similar, but the slopes are much higher. So if you would take the ratio, so that means that clearly the physiological maturation is scaled differently from anatomical maturation. And if you take the ratio of those two, you can come to some sort of developmental scaling coefficient, which comes out, depending on what property you're looking at, between like 2 and 6 times slower development in humans after accounting for these different anatomical growth rates. So there clearly is something that couples physiological maturation to overall brain maturation in mice, which is much less pronounced in humans. So to get to the bottom of that, we did some allometry analysis. This is basically a principle that you find throughout biology, right? That different traits can scale predictably with each other, and they typically form like a linear relationship on a log-log axis. So this is a well-known example for body mass and brain mass. You see across species, there's this linear relationship between the two. Now this also extends to neurophysiology. There was this great paper from the Harnett lab a couple of years ago looking at physiological properties across different species, finding all these different resistances. And underlying that turned out to be this allometric rule that the sort of potassium conductance scales linearly with the total surface area of neurons. Now there's much more to that story, but I'm going to skip over that now. The point is that allometric rules also apply in sort of the physiological domain. And this here are examples like looking at across species, but you obviously also have that within a species as you're maturing, as you're growing. So we looked at sort of within species allometric rules for physiological development and found that in mice, indeed, there are these very strong linear relationships between physiological features and brain volume. So these are just 3 examples, but for mouse that was very consistent throughout all the physiological properties that we were looking at. But that seems much less pronounced in humans. So we've got like a shallower slope, but also the spread around that slope, around that fit is much greater. So there seems to be a strong allometric relationship between the maturation of physiological features and brain volume in mouse, but in humans this coupling is much less pronounced, which may be part of why they can sort of follow such more protracted developmental timelines. So the last bit I want to show you is sort of a bit different. We've seen in recent years that the supragranular layers are very diverse in terms of their transcriptomic types, but also in their physiologies that are present there. So we wanted to understand whether that diversity is something that is there from the start or takes time to develop. So this is a UMAP of 400 cells that we recorded this way of 25 features. And you can see there's a clear progression from infancy to adulthood, basically from the bottom left to the top right of that distribution, of that UMAP space. And so we wanted to see whether we could identify any sort of clusters, developmental clusters, using Gaussian mixture modeling. I wouldn't say we had like very strong, well, there's clearly an infant area there, this E-type 1, then some intermediate sort of groups, and then you end up with this E-type 4 and 5, which really take up all those adult neurons. If you look at the sort of the proportion over time, you see that that infant E-types sort of disappears. Well, you don't really see much in adulthood. And interestingly, this E-type 5 is one that only appears quite late. So if you do this trajectory analysis from our sort of infant cluster to the two adult endpoints, you can see that the divergence of the two paths is very late. And the thing that actually distinguishes the two endpoints of E-type 4 and 5 is this burst firing behavior. So it's measures of spike train regularity. So this E-type 4 is a very regular firing neuron, whereas this E-type 5 is clearly burst firing. And you can see that quite nicely if you look at, for instance, burst index, sort of like how strongly that cell is bursting. You can see that this starts quite late, this increase in the burstiness of these cells. And yeah, the same for local variation, just another metric of like how regular the cells are firing. Another interesting difference is they're exhibiting their SAG ratio. So the layer of these E-type 5 neurons had much stronger SAG ratios than the other types, which was also seen by having a greater sort of resonance than the other E-types. So far, it's looking, this is very similar to the deep T-types that were described in the Verhoog paper, where this strong bursting, strong IH, and this is more anecdotal, but well, the deep location also is confirmed because these were identified more in the deeper layers. But also the few retrieved nicely reconstructible cells we had also sort of lacked those the dendrites in layer 1, which also was remarked upon in the Berg paper as one of the features. So to really get to the bottom of that, I think someone would have to do some patch seek on developing neurons to figure that out. So I'll quickly round off. I think we've seen that the nature of postnatal changes happening between humans and mice is very conserved, but it's clearly on a different time scale, where human neurons seem to mature slower, even accounting for different brain growth rates. And that means that as you're sort of maturing and going through different stages of cognitive development, you're actually doing so with a functionally different population of neurons, not only in their mean values, but also in their variance. And ultimately, that I think also has some implications for the role of synaptic plasticity as you go, as you're developing. Having done a PhD in synaptic plasticity, I always thought that most of the postsynaptic changes in circuitry was always just synaptic plasticity. But to now see that you've got these big changes in intrinsic properties as well really sort of enriches that picture. And I would think that to actually maintain circuit function in the face of having these big changes in intrinsic properties will probably require some a role for plasticity as well, to sort of counterbalance these things. And thanks for your attention. I've got a lot of people to thank, obviously, and just wanted to plant a seed for one day maybe hosting this thing in lovely Cape Town. And thanks for your attention.
Here. Hi. Thank you very much for your talk, Thijs. I have two questions. First of all, did you don't have the data for the synaptic currents, or do you just on purpose didn't include them in this analysis? And second is, so one phenotype that comes very late, like the bursting phenotype at 40, is that maybe already aging pathology, or do you really think it's maturation still at 40 years? Would be nice for me with 40 plus to still mature.
So the first question about the synaptic currents, so synaptic work, we've been working on that, but it's not in a sort of same advanced stage yet. It is quite, this is obviously a more high throughput experiment. You can easily get like a big sample, and if you see how much variation there is, it's probably going to extend to the synaptic side of things as well in the pediatric cortex. So I think it's going to be a couple of years before I present that work. And in terms of the bursting, I think, I don't think it's aging. I don't know. It's difficult to really relate what you see at the synaptic or the neuron level to our own cognitive functioning, but I think that if you look at the intrinsic properties after this sort of cutoff at 55, we cut it off here because that's where we had like nice dense data. After that, the fits would be a little bit more unpredictable with having like more sparse data points. But I don't think much is, I think it's sort of the same pattern is continuing. So you've got a couple of good years ahead of you still, I think.
Thanks, Thijs. Very nice. So you mentioned that you work only in medial temporal gyrus or temporal cortex, which is of course association area. So and you mentioned that the neurons have these intrinsic, protracted developmental time course. Do you think it also would hold for other brain areas like primary sensory areas or primary motor areas, that this intrinsic neuronal time scale is the same as in an association area? Do you think that in the human brain there can be different neuronal trajectories depending on the functionality?
Yeah, I definitely, in the choice for limiting to temporal cortex, I definitely try to sort of prevent different cortexes affecting our data. I definitely think it's a possibility. Although if you look at the sort of, just from like a brain volume perspective, if you look at the different cortical areas, yes, they have different maturation rates, but not that different. Like, for instance, the visual cortex change in size is sort of just maybe a year or two ahead of the temporal cortex, just if you're looking at the MRI scans. But yeah, no, you would probably think that this extreme protraction would be more something for the association areas. I think the more, it's something to look into, I guess. But yeah, we need a big data set, and that's the thing. That's why temporal cortex is also handy. Like, it's very difficult to get like a data set this size from one cortical area alone. So I think for visual, it'll be very much a challenge. Yeah.
Robert.
This is a really great talk, Thijs. I was just curious about the allometric scaling. I've wondered this about the Mark Harnett story too, like, so the sort of lack of strong allometric scaling rule. Do you have any thoughts on, like, is there any sort of advantage to that? Do you have any thoughts on like the functional implication of that? So that's like 2 studies now where you've sort of seen a...
Yeah. So yeah, so in the Harnett story, I think humans were sort of this outlier, right? I sort of glanced over that, basically glanced over the main point of the paper there. Sorry. So you see that red data point, that's humans, really much slower. I thought it was back on the allometric scale if you adjusted for like conductance area. They had some sort of way of transforming it that it would fit again. Yeah, I'm not sure. I think that's I have no theories on why that would be. I can see. Well, you would think that in terms of development, having this, there must be some sort of advantage to keeping your neurons in a sort of this juvenile state for a longer period, which I think ultimately possibly relates back to synaptic plasticity. I mean, I showed you those slow action potentials. I remember a story that was printed last time from, I think, Franz Mittermeier, where there was like, if you depolarize the presynaptic cell, the action potential waveform widened, and that enhanced synaptic plasticity by increasing the amount of glutamate release. You could see something like that happen. So maybe like having these neurons with high resistances for such a long time changes something to their electrotonic structure in a way that sort of is beneficial for learning. But yeah, that's what I can offer. Thanks.